studied the transport of various substances in different portions of the tubules in the last class. We studied that in PCT, most of the ions and all these substances along with urea and other organic and non-organic substances, how they are transported. They are reabsorbed mainly, and 65% of reabsorption of all these substances are taking place in PCT. Coming down to in PCT itself, there are certain substances which are secreted also. Hydrogen ions, ammonium ions, and various other substances, they are secreted in PCT. In coming down to loop of Henle, I told you that the descending limb of loop of Henle is permeable to water. So it just mainly allows just the water to be absorbed here. Then as we go up in the thick ascending segment of loop of Henle, there's a very important ion channel, sodium-potassium 2-chloride channel, which causes major reabsorption of these ions in this particular segment of the tubule. Then DCT also permits sodium chloride ions mainly, apart from bicarbonate, and water always follows these ions. So this is the main channel which is being operated here in DCT. Then there is also secretion of hydrogen ions, potassium ions, and ammonia. We'll be discussing ammonium later when we'll be studying acid-base balance. But these substances are secreted in DCT and in collecting duct, some higher portions of the particle portions of collecting duct as well. And there will be reabsorption of water and urea in the medullary segment of the collecting ducts. This will also be dealt with in detail in the next lecture, when we'll be studying the concentration and dilution of urine. So here we studied this graph as well, where we found out that so many substances, their concentration, they keeps on varying as these fluid passes from the PCT to loop of Henle to distal tubule and then to collecting duct. The glucose, proteins, amino acids are completely reabsorbed, so they are not seen under normal circumstances in these portions of the tubules, that is loop of Henle and further in DCT and collecting ducts. While certain substances which are not reabsorbed at all, inulin, creatinine, pH, etc., their concentration keeps on increasing as the fluid passes further till the collecting duct. While some other substances, they keep on fluctuating and their concentration is in relationship to that of water. So we started with regulation of tubular reabsorption, and there are various mechanisms which regulate the tubular reabsorption. The first and foremost is the glomerular-tubular balance. Then there are various peritubular physical forces which also work, and we'll be going into detail of all these forces. We'll be going into detail of all the hormonal factors which influence tubular reabsorption, and then other factors like sympathetic nervous system, arterial blood pressure, and osmotic factors which help in regulating the tubular reabsorption. So we'll be going one by one and studying in detail how these factors influence the tubular reabsorption. So tubular reabsorption, the first mechanism is the glomerular-tubular balance. It just explains the direct proportionality of the tubular reabsorption as compared to the tubular load. That means whatever amount of load is received by the tubules depends on the GFR. So whatever amount of load is received by the tubules is directly proportional to the tubular reabsorption. Okay, so it increases. Once the load increases, the tubular reabsorption also increases. So it is a form of auto-regulation. And it can explain the intrinsic ability of tubules to increase their absorption rate in response to increased tubular load. So that means the tubules have that ability to increase their absorptive rate whenever there is increased load given to them. And this total reabsorption increases, but the fraction of this reabsorption remains same. That means if 65% is being reabsorbed from PCT, that fraction of 65 will remain same. And it helps in preventing the overloading of DCT when GFR increases. And I have given you this example, and I'll be explaining this example, how this is helping. So the importance of glomerular-tubular balance in minimizing changes in the urine volume is depicted with this example. Here what do we observe is, under normal circumstances, the GFR is 125, isn't it? And the reabsorption, as I told you, is 99%. Total reabsorption from the tubules is 99%. We are not discussing PCT right now. The complete reabsorption which takes place is 99%. And the urine volume is just 1 liter. So reabsorption, if it is 99%, it will be about 125, 124. So if there is no glomerular tubular balance in the kidneys, and if the GFR increases from 125 to 150, what will happen? If the same amount of substances are being reabsorbed, that is 124, are being reabsorbed, the reabsorption will be just 82%, 82.7%, and the urine volume will increase to 26 liters. So you can imagine what will happen. But under normal conditions, this is not occurring. And with perfect glomerular tubular balance, with increase in GFR to 150, 99% of substances will still be reabsorbed, so that the reabsorption increases to 148.8 and the urine volume just increases to 1.2 liters. So this is the role of glomerular tubular balance in maintaining the volume of urine. So that is a very important autoregulatory mechanism taking place at the level of tubules. Okay, so here I'll again explain that if GFR increases from 125 to 150, and if there is no glomerular tubular balance, the same amount of substances will be reabsorbed and the urine volume will increase tremendously, which will be hazardous. But if there is perfect glomerular tubular balance, that means the same 99% is being reabsorbed, then the amount reabsorbed will increase proportionately and the urine volume will only slightly increase, which can be managed. Coming to the next factor, which is altering the tubular reabsorption, and that is peritubular capillary reabsorption. We all know that these are the tubular cells, and various changes, various reabsorptive mechanisms are taking place at the luminal border of these cells and also at the basolateral border of these cells, isn't it? But what happens after this? Once it reaches, all these substances reaches the interstitial tissue, it has to be transported to the peritubular capillaries. So how these are getting reabsorbed in the peritubular capillaries, that is dependent upon the various Starling's forces. We have studied all these Starling's forces in GFR. So same applies here. The Starling forces, that is the peritubular capillary hydrostatic pressure, plasma oncotic pressure. Similarly, these pressures, hydrostatic pressures and oncotic pressure in the interstitial fluids. So these will try to transport the substances here and there accordingly, because we know that if there is increase in the hydrostatic pressure of the peritubular capillaries, the fluids will move in this direction. So there will be no reabsorption in that case. While if oncotic pressure of the peritubular capillaries increases, they're going to pull the fluid towards them. Okay, so now I think you know all the hydrostatic forces. We have already studied in detail in GFR. So all these forces will act here, and the net absorption pressure, if we calculate this, it comes out to be 10 millimeter mercury in the direction of the capillaries. So that will be the bulk flow. The water will be carrying all the substances along with it, which are reabsorbed in the interstitial tissue, and they will be moving inside the peritubular capillaries. Again, in the peritubular capillaries, the net reabsorption, the reabsorption, total reabsorption will be equal to net reabsorption pressure, which we have studied just now, and also the filtration coefficient, similar to what we have seen in GFR. These two factors regulate the transport. So net reabsorption pressure or net filtration pressure, which we had observed in the last slide, that was 10 millimeter mercury, and the filtration coefficient, which is this. And filtration coefficient, we all know it depends upon the surface area and the permeability of the membrane. So this is the net reabsorption, which is observed in the from the interstitial tissue to the peritubular capillaries. So determinants of peritubular capillary reabsorption is filtration coefficient. If this is increased, what will happen to reabsorption? We cannot be interactive right now, but if it would have been the class, you will be answering all these things. So just remember what you just please be keep thinking what happens if there is increased filtration coefficient. The reabsorption will obviously increase. If there is more hydrostatic pressure in the capillaries, reabsorption will decrease, and if there is more oncotic pressure in the capillaries, reabsorption is going to increase. So coming to the hormonal role of various hormones in causing reabsorption of various substances. So first and foremost is aldosterone. As we all know, it acts on late distal tubules, cortical, and medullary collecting tubules. So these are the sites where aldosterone acts. And in these areas, there are two types of cells, principal cells and intercalary cells. So principal cells causes increased sodium reabsorption. Under the influence of aldosterone, sodium gets reabsorbed. And it increases potassium secretion also. So the main role of aldosterone is to reabsorb sodium and to secrete potassium. Additionally, it also increases hydrogen secretion from the intercalated cells. Okay, so the major role of aldosterone shown here is it is reabsorbing sodium, secreting potassium. That is a major role, and that it is doing by acting at both the places. First place, you can see that at the basolateral surface there is sodium potassium ATPase, so it is stimulating this pump. On the other hand, it is also causing the passive movement of sodium because now the gradient has been developed due to this pump. So it is also helping or stimulating this passive transport of sodium here. So at both the places, aldosterone is helping and causing the movement of these ions. And on the other hand, this also, because of the pump, the potassium enters inside the cell and it will be secreted out. So these are the major role of aldosterone in the collecting tubules and in the late distal portion of the tubule. So if there is abnormal aldosterone production, like when there is excess aldosterone, as happens in primary aldosteronism or COM syndrome, what will happen? More sodium retention will be there because it's the main role of aldosterone, as we discussed, is retention of sodium and secretion of potassium. So in condition when there is excess aldosterone, there will be sodium retention and hypokalemia. That means less amount of potassium in the blood is hypokalemia. There will be alkalosis, as we discussed earlier, that increase in the activity of sodium hydrogen exchange also takes place and it is secreting hydrogen also. So that it will lead to alkalosis. And the retention of sodium always causes increase in blood pressure, that is hypertension. So these all are the features of primary aldosteronism when there is excess aldosterone secretion, and the main action of aldosterone is retention of sodium, secretion of potassium, secretion of hydrogen ions. So all these features will be observed in this condition. On the other hand, if there is aldosterone deficiency, this happens in Addison's disease. There will be loss of sodium, there will be hyperkalemia, that means increased potassium levels, and hypotension. The opposite conditions will occur. I hope you understand this. If you again, I will repeat, aldosterone, the main role of aldosterone is to reabsorb sodium and to secrete potassium. So when there is excess of aldosterone in any condition, it causes sodium retention and hypokalemia, along with alkalosis because hydrogen is also secreted, secondarily leading to hypertension. While if there is deficiency of aldosterone, there will be loss of sodium, hypokalemia, and hypotension. So how is this controlled? The factors that increase aldosterone secretion are angiotensin II. This also we discussed earlier in renin-angiotensin system, that once angiotensin is formed, it leads to increase in the secretion of aldosterone. So first thing which controls aldosterone secretion is angiotensin II. If there is increased potassium levels, that also increases aldosterone secretion. And the ACTH, adrenocorticotropic hormone, which is liberated from anterior pituitary, that also acts on the adrenal cortex and causes liberation of adrenal cortical hormones. One of them is aldosterone. Factors that decrease aldosterone secretion is atrial natriuretic factor. As the name indicates, this factor is liberated
Muted.
And it is natriuretic. That means causes excretion of sodium ions in the urine. Okay, so atrial natriuretic factor, and it is liberated whenever there is stretch of the walls of atria, and once this is liberated, it causes natriuresis, that is excretion of sodium in the urine. So that factor also decreases aldosterone secretion, and increased sodium concentration, if it is there, which is increases molality, that also causes decrease in aldosterone secretion. So coming to angiotensin II, angiotensin II increases sodium and water reabsorption. And how it is doing that? It is stimulating aldosterone secretion. We have already seen this, that angiotensin 2 stimulates aldosterone secretion, which in turn causes sodium reabsorption and secondarily water reabsorption. Second mechanism of angiotensin 2 is it also directly increases sodium reabsorption from proximal tubules, distal tubules, loop and collecting tubules. It constricts efferent arterioles. This we had studied in the lecture of GFR, how angiotensin 2 has got a preferential role on efferent arterioles, causing its constriction. And once the efferent arterioles gets constricted, it decreases peritubular capillary hydrostatic pressure, because now no more blood is flowing, or very less amount of blood is flowing to the peritubular capillaries. When the efferent arterioles gets constricted, the blood flow to peritubular capillaries will be decreased. So there will be decreased hydrostatic pressure in the peritubular capillaries. So what will happen? There will be increased filtration fraction. This happens in the filtration fraction which we see. And I hope you remember what is filtration fraction. It is GFR upon the renal plasma flow. That is going to increase. And this increases peritubular colloid osmotic pressure. Okay. So these mechanisms are the mechanisms by which angiotensin is producing its effect and thus increasing the sodium and water reabsorption. So this angiotensin 2 shown here, it has got its receptors located at the basolateral surface as well as on the luminal surface of the cell. And once angiotensin acts on these receptors, it stimulates sodium potassium pump first of all, and also sodium bicarbonate O transporters. These two are stimulated at the basolateral surface of the cell. While on the other hand, it is also stimulating sodium hydrogen antiport on the luminal border of the cell. So all these things are stimulated by angiotensin 2. So again, angiotensin 2, when it acts on its receptors, it activates certain mechanisms which helps in or stimulates sodium potassium ATPase activity. It also stimulates the co-transport of sodium and bicarbonate ions. Secondly, on the luminal surface, again this angiotensin, when it acts on its receptors at the luminal surface, it is stimulating the antiport of sodium and hydrogen and thus helping in secretion of hydrogen ions, in return reabsorption of sodium ions. So this is how it is reabsorbing the sodium. So this is the same thing which we discussed, that how angiotensin 2 preferentially acts on the efferent arteriole. This is the glomerular shown here, this is afferent arteriole shown here, and this is the efferent arteriole shown here, and this is the peritubular capillary. So this is showing the constriction of the efferent arterioles. So when this is constricted, which happens with angiotensin 2, there will be decrease in peritubular capillary pressure. Obviously, the blood flowing till here, it is very high pressure, the blood is, but here there is constriction, the pressure here will be less. So peritubular capillary pressure decreases. So this will decrease the renal blood flow, increase filtration fraction, and increase oncotic pressure here. Okay. So again, when there is sodium depletion, there will be increase in angiotensin 2 concentration, which increases the resistance of efferent arterioles. It acts by various mechanisms. First, increases glomerular capillary pressure and thus prevents decrease in GFR and retention of waste products. Again, this increased efferent arteriolar resistance decreases renal blood flow. And when there is decrease in the renal blood flow, the filtration fraction is increased, because filtration fraction is GFR upon renal blood flow. So when renal blood flow is decreased, filtration fraction is increasing. At the same time, decreased peritubular capillary pressure is also decreasing. So when peritubular capillary pressure is also decreasing, these two factors combined will cause increased reabsorption of sodium and water from the tubules. Okay. So again, I'll repeat the conditions which increases the reabsorption of sodium and water due to angiotensin 2. Angiotensin 2 causes increased resistance of efferent arterioles preferentially, although it constricts both the arterioles, but preferential role is on the efferent arterioles. So it decreases glomerular capillary pressure and thus prevents the decrease in GFR. It decreases renal blood flow, decreases peritubular capillary pressure, thus increasing filtration fraction and thus increasing sodium and water reabsorption. So what will happen if there is blockage to angiotensin 2? There will be decreased reabsorption of sodium and blood pressure will also be less. So there are certain drugs, substances given to produce this effect, like ACE inhibitors. By ACE we mean angiotensin converting enzyme inhibitors. These are certain drugs given here. They come under the category of ACE inhibitors. Okay, so we can give this because what is ACE doing? Angiotensin converting enzyme is converting angiotensin 1 to angiotensin 2. So if we give this inhibitor, angiotensin 2 will not be formed. Secondly, we can give angiotensin antagonists or renin inhibitor. So all of these will function similarly because ultimately it is renin, angiotensin, aldosterone pathway basically. So renin angiotensin system will be affected here. Either we give the blockade of inhibitor of renin or antagonist of angiotensin 2 or ACE inhibitor. This is commonly given. So what all these do? They decrease aldosterone secretion, they directly inhibit sodium reabsorption, and they decrease the efferent arterial resistance. And thus they cause natriuresis and diuresis. Natriuresis, as I told you, is increased excretion of sodium in urine, and diuresis is increased water in urine. That means the increased amount of urine. Okay, and ultimately causing decrease in blood pressure. So these are very important drugs given to control blood pressure in people having hypertension. ACE inhibitors are commonly given. And with this role, ACE inhibitors, by decreasing the liberation of aldosterone, they're decreasing the reabsorption of sodium and thus favoring natriuresis and diuresis and ultimately controlling the blood pressure. Atrial natriuretic peptide also increases sodium excretion. Now, atrial natriuretic peptide, as I already told you, that atrial natriuretic peptide, as the name indicates, it is secreted by cardiac atria. That is, atria is liberating it in response to stretch. So when the stretch occurs when there's increased blood volume. So when there's increased amount of blood, increased blood volume is reaching the atria, there will be stretch in the atria. There are stretch receptors present in atria, so they get stretched, and due to this particular substance ANP, that is atrial natriuretic peptide, is liberated and it directly inhibits sodium reabsorption. It also inhibits renin release and aldosterone formation, increases GFR, and thus it helps to minimize blood volume expansion. How it is happening is shown here in this flow diagram. Whenever there is increased blood volume, there will be increased secretion of atrial natriuretic peptide from atria. This decreases renin release, decreases aldosterone. Renin causes decrease in angiotensin 2, causes decreased renal sodium and water reabsorption. Both these things, angiotensin 2 and aldosterone, are doing causing actually sodium and water reabsorption. So when these are decreased, the renal reabsorption of sodium and water will also be decreased. So there will be increased excretion of these substances, ultimately managing the blood volume, decreasing the blood volume. So that is the role of atrial natriuretic peptide. So how this is a very important mechanism shown here, and that how aldosterone, due to aldosterone infusion, actually there should be retention of sodium. Just now we have seen that aldosterone is causing retention of sodium. But after some time, it is seen that aldosterone is not able to retain sodium. So this is known as escape phenomena. That is escape from sodium retention during excess aldosterone infusion. If we give aldosterone infusion, what will happen is initially the urinary sodium excretion will decrease. As you can see in the lower graph here, the renal sodium excretion decreases for some time. Okay, for a very short time, it decreases. But suddenly what you observe is that with continuous aldosterone infusion, this escapes and the urinary sodium excretion becomes normal. So this escape phenomena can also be explained because once this aldosterone is causing sodium retention, there is secondarily retention of water, and due to the stretch, the ANP is liberated, causing increased excretion of sodium, that is natriuresis and diuresis. Okay, so these two hormones will be interrelated, and that will cause the escape from escape phenomena, which is quite often observed in aldosterone infusion. So initially, I'll repeat this phenomena here. Initially, when we give aldosterone infusion, first thing which will happen is initially aldosterone will produce its effect, causing retention of sodium. When there is retention of sodium, less amount of sodium will be excreted. That is causing retention of sodium. But very soon, continuous infusion of aldosterone will cause escape phenomena, and sodium, the concentration of the amount of sodium excreted will become normal. This happens because sodium, once retained, causes retention of water, causes ANP stimulation, that is atrial natriuretic peptide, which causes natriuresis and diuresis and maintains this excretion of sodium. Coming to the next hormonal control of the tubular reabsorption, and the next hormone is antidiuretic hormone. We discussed this also at various places. So what is ADH doing? We all know that ADH is secreted from posterior pituitary, and it increases water permeability and reabsorption in distal tubules and collecting tubules. It allows differential control of water and solute excretion, and is an important controller of extracellular fluid osmolarity. ADH, you can see in this figure, ADH is synthesized in the magnocellular neurons of hypothalamus. If you remember supraoptic nucleus and paraventricular neurons, so these are the neurons, supraoptic and paraventricular neurons in the hypothalamus. And these neurons, they travel to the posterior lobe of pituitary and release ADH here in the posterior lobe, in the posterior pituitary. And this is liberated from here, released from here, goes to the kidneys and produces its effect in the collecting tubules and distal portion of the TCT also. So these are the places where ADH, antidiuretic hormone, is liberated, and as its name indicates, it is antidiuretic. So it prevents diuresis. Okay, antidiuretic hormone will prevent diuresis. So it will cause reabsorption of sodium and water and prevent the diuresis. How does it cause this effect? It is explained in this figure. I hope you remember the other name of ADH. What is antidiuretic hormone also known as? It is known as vasopressin, or we can say arginine vasopressin. AVP written here is arginine vasopressin, which is another name of ADH. Okay, so you can see on the basolateral surface of the renal tubular cell, there are receptors present for ADH to act. So these are the receptors. These are G protein coupled receptors, you can see. Okay, so whenever these ADH or arginine vasopressin, it binds to these receptors, G protein receptors are there, they get stimulated, ATP gets converted to cyclic AMP, and this mechanism you have already studied in the hormonal action, mechanism of hormone action. Many hormones act by this mechanism. So you have already studied there. You must have studied in detail in biochemistry also. So one of the mechanisms by which the hormones act is this. When the G protein gets stimulated, ATP gets converted by adenylate cyclase pathway. This is known as the ATP gets converted to cyclic AMP, which causes protein kinase A formation, which causes phosphorylation of proteins, and this causes new protein formation. And these new proteins which are formed are aquaporin molecules. So this is aquaporin 2 which is formed. They migrate from here. Once they are formed, they migrate and they get exocytosed and gets accumulated at the luminal border of the cell. Okay, so they get attached by means of exocytosis. They get attached here to the luminal surface, and once aquaporin 2 molecules get attached here, they cause entry of water inside the tubular cells. So mainly the role is that of aquaporin 2 on the luminal surface to cause reabsorption of water, although it is observed there are aquaporin 3 and 4 also, which are present at the basolateral surfaces. But the main action of ADH is on the luminal border, where it causes reabsorption of water by synthesizing aquaporin 2 molecules by this mechanism which I explained here. I hope it's clear. The aquaporin, how they are formed by the ADH, by the G protein mechanism, by activation of adenylate cyclase, causing the synthesis of new proteins, which gets attached to the luminal border. And once they get attached to the luminal border, they cause the channels to open, the water channels. The aquaporins are nothing but water channels, and water gets reabsorbed. So whenever there is extracellular osmolarity, it increases, more amount of ADH will be secreted, increased tubular water permeability will occur, more water reabsorption will occur, decreased water excretion will occur, and that will manage the increased extracellular osmolarity. The same mechanism, the ADH mechanism, is explained here in a way of flow diagram, that how ADH secretion causes reabsorption of water and decreases the extracellular osmolarity. So when there occurs abnormalities of ADH, as is depicted here, like one of the condition when there is excess ADH secretion, inappropriate ADH syndrome, also known as SIAD, syndrome of inappropriate ADH secretion. So when there is excess ADH secretion, there will be decreased plasma osmolarity and hyponatremia. Why? Because more amount of water will be reabsorbed, and more amount of water being reabsorbed decreases the plasma osmolarity. On the other hand, in central diabetes insipidus, when there is insufficient ADH secretion, there will be increased plasma osmolarity, hyponatremia, and excess thirst. Okay, so these are the abnormal conditions when there is either excess ADH secretion or insufficient ADH secretion. What would happen? So we can easily explain now. Whenever there's increased ADH secretion, water will be more reabsorbed, so there will be decrease in the osmolarity. While when there's insufficient ADH secretion, less amount of water can be reabsorbed, there will be increased plasma osmolarity, hyponatremia, and excess thirst. Now coming to parathyroid hormone, and its main role is in reabsorption of calcium. It is released by parathyroids in response to decreased extracellular calcium. So whenever the extracellular calcium is less, parathyroid hormones will be hormone will be secreted, and it increases calcium reabsorption by kidneys. It also increases calcium reabsorption by gut and decreases phosphate reabsorption. So that is the main role of parathyroid hormone. It is causing increased reabsorption of calcium and decreased phosphate reabsorption. And thus it helps to increase extracellular calcium levels. So whenever there is decrease in extracellular calcium, there will be increased PTH secretion, parathyroid hormone secretion. On one hand, it increases vitamin D activation. It causes vitamin D activation, which increases intestinal calcium reabsorption. On the other hand, parathyroid hormone increases renal calcium reabsorption, and it also causes calcium release from bones. So parathyroid hormone causes increase in plasma calcium level at all the three levels, right? That finishes with the hormonal regulation of tubular reabsorption. Now coming to the role of sympathetic nervous system. The sympathetic nervous system directly stimulates sodium reabsorption. First of all, it also stimulates renin release, and it decreases GFR and renal blood flow. Only a high level of sympathetic stimulation causes decreased GFR and renal blood flow, mainly because it is causing constriction of the arterioles, and constriction of the arterioles decreases the GFR and the renal blood flow. So that is its role. But it is also directly stimulating the sodium reabsorption. Now, what is the role of increased arterial blood pressure? If there is more blood pressure, it decreases sodium reabsorption, and this is known as pressure natriuresis. So first of all, what is pressure diuresis? That is when there is increased blood pressure, it causes a rise in urine volume. There will be increase in the urine volume excretion. And pressure natriuresis is rise in sodium excretion that occurs with elevated blood pressure. So whenever there is increase in blood pressure, there will be pressure diuresis and pressure natriuresis. That means there will be increased excretion of both sodium and water. How it is happening? There will be increased peritubular capillary hydrostatic pressure. There will be decreased renin and aldosterone. Increased release of intrarenal natriuretic factors will also be there. So all these mechanisms together will cause increased excretion of sodium and water. Okay, there will more and more be secreted, and less will be reabsorbed. So what are the osmotic effects on reabsorption? As we know that water is absorbed only by osmosis. So if we increase the amount of unreabsorbed solutes in the tubules, there will be decreased water reabsorption. That means the solutes present in the tubules, if the amount of these solutes will increase or less amount of solutes are being reabsorbed, then water will also not be reabsorbed. What happens in diabetes? There is so much glucose. There's increased glucose levels. And when it is uncontrolled diabetes, so much glucose is filtered out in the tubules that it exceeds its capacity of reabsorption. So in tubules, it's unreabsorbed glucose, the amount of unreabsorbed glucose, it increases in the tubules. And that causes diuresis and water loss, because now water will not be absorbed to that amount, because due to the glucose, it produces osmotic effect and retains water in the tubules, and water will be lost. That's why we see polyuria in diabetes mellitus. If you remember, if you have studied, when you studied diabetes, you must have remembered how hyperglycemia, polyuria, polydipsia, all these things occur. So they are all interrelated, because so much amount of glucose is there in the tubules which is not being reabsorbed. So that leads to osmotic effect, and the water will also be retained in the tubules, leading to water loss. So this is osmotic diuresis, osmotic diuresis, which is observed in diabetes. Another thing, we can give certain substances like mannitol. They also produce the osmotic diuretic effect. Then coming to the abnormal tubular functions. Now I'll be telling you some functions, some conditions in which the tubular function becomes abnormal. Like renin secreting tumors. Whenever a tumor, some tumors are there which secretes renin to a very high extent. So what they do, they increase angiotensin 2 levels, of course, and increase aldosterone levels. So we all know the effect now very well, what happens when renin, angiotensin, aldosterone system gets activated. You can yourself think over what happens to sodium reabsorption. It increases. So what happens to sodium excretion? It almost remains, it tries to balance it and tries to make it controlled. There will be increased potassium secretion. Maintaining of potassium excretion will also occur, but potassium plasma, potassium level decreases. There will be rise in blood pressure. There will be increase in aldosterone level. Okay, so you can just relate, just think over the renin-angiotensin system, which you have studied many times now, and think what is the main role, how it is reabsorbing sodium and all, and how these effects will occur. Coming to one syndrome known as Liddle syndrome, it occurs when there is excess activity of amiloride-sensitive sodium channels and late distal and cortical collecting tubules. We have studied these channels in these tubules, cortical collecting tubules, which there were channels in these tubules which were causing entry of sodium or reabsorption of sodium. So when there is excess activity of these channels, then this syndrome, Liddle syndrome, occurs. And therefore, sodium channel blockers like amiloride and triamterene, these are sodium channel blockers. This, when given, will help in treating this particular symptom. And since these channels are sensitive to this drug amiloride, these are known as amiloride sensitive sodium channels, right? So when we were discussing the reabsorption of substances in the late distal tubules and in the cortical collecting ducts, I told you about these channels where sodium is being reabsorbed. So when there is excessive activity of these channels, so much amount of sodium will be reabsorbed. So this happens in Liddle syndrome, and the certain drugs which block these channels like amiloride, they can be given. They will block this channel and can help in treating this condition. So there is in Liddle syndrome excess sodium channel activity in late distal tubules and collecting ducts. So sodium reabsorption increases, sodium excretion will be in steady state, BP increases, plasma renin decreases, and aldosterone decreases. These are the features observed in Liddle syndrome. Coming to another syndrome associated with some other channels, and these channels are sodium-potassium 2-chloride channels. These are co-transporter of these three ions, which we have studied in thick ascending limb of loop of Henle. I told you in thick ascending limb of loop of Henle, this is a very important transport channels which are there, which are acting, and this function of these channels will be further emphasized in my next lecture when we will be studying the concentration and dilution of fluorine or countercurrent mechanisms. So these are very, very important channels. They transport one sodium ion, one potassium ion, and two chloride ions at the same time. So these channels, their activity is blocked in case of Bartter syndrome. Their activity is decreased in Bartter syndrome. So these substances will not be absorbed. So the treatment will be high NaCl and potassium intake will be done, and various other things which are not very significant. You can leave this part, but you just remember that in Bartter syndrome, this channel is inhibited. In a way, it is inhibited. So these substances, important substances, sodium, potassium, chloride, cannot be transported inside the cells. In Gitelman syndrome, there's decreased NaCl reabsorption in early distal tubule. I told you in early distal tubule, there is a co-transporter of sodium and chloride. So when this is inhibited, these substances cannot be reabsorbed. That is in Gitelman syndrome. So this is the channel, sodium chloride co-transporter, this channel which is present in the early distal convoluted tubules. When the activity of this channel gets decreased, then these substances will not be transported at this level. That is Gitelman's syndrome. So here also high NaCl and potassium intake needs to be done along with some magnesium supplements, because magnesium also gets transported at this level. So that will also be altered indirectly. Another syndrome is Fanconi syndrome. Here there is generalized decrease in reabsorption in proximal tubules. So we all know that how much reabsorption is taking place in the proximal tubules. 65% of reabsorption of various substances. You can see sodium, phosphate, bicarbonate, glucose, amino acid, all the substances, they are reabsorbed here, and majority is taking place in proximal tubules. So if there is decrease in this reabsorption, there will be a major changes. There will be phosphaturia, renal glycosuria, amino aciduria, because then these substances will not be absorbed that much in the other portions. Major absorptive capacity is that of PCT. So they will be, their levels will be increased in the tubules. So they will come out in urine. So amino aciduria, glycosuria, tubular proteinuria, and proximal renal tubular acidosis, because hydrogen is also secreted in this tubule. So that will also not be there. So there will be acidosis as well. Causes, it can be genetic, it can be due to heavy metal damage or due to certain drugs, multiple myeloma, tubular necrosis, the number of causes. Even if you don't remember, that's not that important, but you should know what is Fanconi syndrome and where it is, the reabsorption is hampered at PCT level. Renal tubular acidosis, when there is decreased hydrogen secretion, there will be increased bicarbonate excretion and acidosis. Now, someone asked me that why increase bicarbonate excretion when bicarbonate is being reabsorbed at various places of the tubules. Whenever the reabsorption of bicarbonate is hindered, it will be excreted. And this will happen in conditions to maintain the pH. Okay, so in renal tubular acidosis, there will be increased bicarbonate excretion and there will be acidosis due to decreased hydrogen ion secretion. So, to summarize all these diseases which we have studied, proximal tubule, Fanconi syndrome, here the absorption of substances are not taking place. Thick ascending limb, Bartter syndrome, sodium potassium chloride is not being reabsorbed. Then Gitelman and Leigh syndrome here in distal convoluted tubules, and there is pseudo hypoaldosteronism. This it is hypoaldosteronism. So when there is hypoaldosteronism, it causes decrease in the reabsorption of sodium. So the proteinuria is the first sign of renal disease, you can say, and it is the early detection of renal disease in at-risk patients, and it is usually observed in cases of hypertension, diabetes, in certain cases of pregnancies like gestational proteinuric hypertension, or we say a complication of pregnancy is preeclampsia, and that also protein appears in urine. And during annual checkup, actually it is essential to estimate protein in urine because renal diseases can be silent. Only the detection of protein will give us an indication of renal abnormality. So it is very important to assess and monitor the protein levels and of known renal disease also, and otherwise also whenever during pregnancies and in certain conditions, if urine albumin appears or protein, any kind of protein appears in urine, it is an indication of renal abnormality. Although it is commonly seen in these conditions like hypertension, diabetes, pregnancy, etc. That's all for today. So, regulation of reabsorption in the renal tubules. I hope you are understanding the lectures. And I'll just see the chats, whether there are any questions. I'm always uploading the PPTs on Google Classroom, and I will upload it today itself. So please don't bother about PPTs. Some people always ask for PPTs, and I'm regularly sending them on Google Classroom. Please approach the Google Classroom and download or see the PPTs there. It is there. Now, someone is asking about SIAD. What is SIAD? It is syndrome of inappropriate ADH secretion. A syndrome of inappropriate ADH secretion means there is excess of ADH secretion. So just think over whenever there is excess of ADH secretion, what happens whenever there is excess of ADH secretion, antidiuretic hormone secretion, the water is antidiuresis. It is causing antidiuresis. So diuresis is not happening, water is getting reabsorbed. Please explain protein diuresis and natriuresis. Protein diuresis and natriuresis. See, when there is increased proteins in the urine, osmolarity of the urine is going to increase. And when there is osmolarity of urine going to increase, more amount of water will come out in the urine, and more amount of, along with water, some amount of sodium will also come out. So that will be diuresis and natriuresis. Please explain absorption in peritubular capillary slide again. Okay, I'll just go to the slides. First, I'll read all the chats and then I'll go back to the slides for explaining these. See, if someone is concentration in one of the graphs I've shown, please, this will be explained in detail in renal function test. Another lecture, separate lecture will be taken on renal function test, where we will be telling you how inulin and creatinine and all these substances, they help in the determining GFR or other things. So just wait till then, we'll be explaining this in detail in a separate lecture. So some of you, the questions which are asked, I mean, they will be covered in the next few lectures. So acid-base balance questions and all those questions of the acid-base balance or of concentration and dilution of urine, that will be covered in the next lecture. And this thing also, creatinine and inulin, that will be covered in renal function question. Why acidosis in Fanconi's cause tubular reabsorption is taking place in the proximal tubules, and even hydrogen secretion, that is also affected. So when hydrogen is not secreted, that will lead to acidosis. What causes osmotic pressure of 15 millimeter mercury in interstitial fluid? So many substances are being reabsorbed. from the lumen to the cells and from cells to the interstitial tissue. So obviously osmotic pressure will be there, and due to reabsorption of all these substances, there is osmotic pressure. Someone is asking how high protein intake increases both renal blood flow and GFR. This I had explained very well in my last lecture by showing a flow diagram, how high protein intake increases both renal blood flow and GFR. Someone is asking that test results and answer key of first test of 28th May. What is 28th May? I'm unable to understand. I don't think this date has still arrived. So we'll wait for this date to come. Then we will have a test and then we'll do the results. So some date mistaken. And I have already uploaded the key of first test of renal physiology. It is there on the Google Classroom. And I think it has been given to your class group also, WhatsApp group. So please check it again. You will get that key of the renal physiology test. Okay. Now coming back to the slides. Someone has asked which slide? Of peritubular capillary slide. Go to that slide. Yeah, so how peritubular capillary reabsorption is taking place and how this osmotic pressure is developed here. See, it's very clear that substances are reabsorbed from the lumen into the cell and from the cell into the interstitial fluid. So whatever osmotic osmolarity is being developed here is due to these substances. Okay. So these substances which are reabsorbed, that rises the osmolarity, and these are the pressures, same Starling's forces which we have studied in GFR. There is hydrostatic pressure in the capillaries, there is osmotic pressure in the capillaries, and this is very high due to plasma proteins. Plasma proteins are there in the blood, so that increases the osmotic pressure. There is hydrostatic pressure, so hydrostatic pressure will try to move the fluid in this direction. Oncotic pressure will try to reabsorb substances. Okay, so we see the balance of these forces. Similarly, there will be some amount of hydrostatic pressure here in the interstitial fluid and some osmotic pressure, because so much of water and so much of osmotically active substances are being reabsorbed. So if we see the balance of all these forces, there is a net reabsorption force of 10 millimeter mercury that causes the movement of fluid from the interstitial tissue to the peritubular capillaries. I hope that clears your doubt. And this reabsorption is not only dependent upon the net reabsorption pressure, but also on the filtration quotient, which is dependent on surface area and permeability. This also I had explained. Now please read these lectures and do it regularly. We will be taking regular tests. I hope the schedule is there with you. Even if it is not there, do it regularly. We will be giving you the notices of the test and we will be taking tests regularly. If any question is left, please write it to me on my email. You can ask there and I will be replying all these questions. While some questions which people had asked in the last class also, I had told them that please wait for the further classes. I will be discussing the concentration and dilution of urine, and we will be taking classes on acid-base balance, on renal function test, where clearance will be taught in detail. So these will be taken later on, so I'm not going into answering those questions. That will be taken later. Okay, so that's all. I hope you all are fine. Please continue to